All right, welcome back. We're continuing our conversation on chapter 3.3, which is genetics and heredity. We're going to be moving on to part 3, which is called Beyond Mendel's Laws. Now, before we talk about that, a quick review on what we've already discussed. In part 1 notes, we talked about meiosis, which is a type of cell division that results in the formation of gametes. Gametes, or sex cells, are especially important for sexual reproduction because they are haploid, meaning that they have half the genetic material, so that when gametes from two different parents join together, that's how you get the full genetic code, the diploid cell. And this allows for genetic variation as well as genetically unique offspring. And then in part 2, we talked about some of the history of genetics as well as how to do Punnett squares. Now, in terms of the history, we mentioned Gregor Mendel, who is an Augustinian monk, and he's considered the father of modern genetics. The reason why he's considered the father of modern genetics is because through his work with the pea plants, he was able to figure out how genes get passed on, specifically what we call single gene inheritance or Mendelian genetics. How one gene affects one trait and how there are autosomal dominant and recessive versions. Autosomal just means not sex linked, which we'll get to that in a little bit. But in terms of what he studied, he was looking at an organism, the pea plants, and the thing about them is they have very simple modes of inheritance. So they generally only have two alleles, two versions of the gene, and it's either dominant or recessive alleles. But what we've found out since then is that it's not always as clear cut as that. What I mean by that is with humans, since that's what we are, very few of our genes, of our traits, are coded for with single gene inheritance. There's different modes of inheritance that Mendel never even knew about through his study with pea plants. And that's why we say that he did pave the way and he laid the foundation for modern genetics. But we know a lot more now than he did then. And that's what it is with any field of study. Someone starts it off, and then as the years go by, we learn more, technology gets better, we learn even more. So it kind of makes sense that, over or almost 200 years later, we know a lot more than he did then. So let's start talking about some of these modes of inheritance that go beyond Mendel's laws. So the first one we have is called incomplete dominance. So you should know incomplete means not complete or not fully. And what this means is you can have two alleles, but neither one is dominant. They're not true recessive either, but they're also definitely not dominant to each other. As a result, what you end up seeing is, since neither one is dominant, neither one is going to be fully expressed. And what that means is then if you have a heterozygous individual, it's going to show a blending of the traits, some kind of intermediate. As you can see on the picture, certain flower species, their petal colors is determined by codominance. And here we have an individual that is homozygous for the red allele. So they have two red alleles. Over here, we have an individual who is homozygous for the white allele. So they have two white alleles. If you cross those two plants, those two flowers, you don't get a red offspring, you don't get a white offspring, you get this right here. The heterozygous individuals or offspring of this cross end up blended. And what happens when you blend red and white? You get pink. So we get this intermediate where it's neither this nor that. It's something else completely. So it's not red like one parent. It's not white like the other. It is pink. We take the traits and we blend them together to get this intermediate, this in-between expression. So the genotypes, homozygous red crossed with homozygous white gives us heterozygous. The phenotypes, red, white, gives us pink. All right. Codominance. Now, the prefix co means both, with, etc. So in codominance, you have alleles that are both dominant. And as we keep saying, a dominant allele must be shown. So what that means is that with codominance, we actually see both traits. So both of the expressions are equally seen. If you look at the examples below, we have here this flower. We mentioned previously that some species of flowers have incomplete dominance as their mode of inheritance for petal color. However, there are also others that show codominance, and that's what we see here. So a homozygous red flower crossed with a homozygous white flower, and what you get in the offspring is going to be heterozygous with one red allele and one white allele. But as you can see in this picture, we end up seeing both colors. So red and white come together to make a flower that has portions that are both red and portions that are both white. So that's different than what we saw with incomplete dominance. With incomplete dominance, since neither allele was dominant to the other, neither one was expressed all on its own, which is why they blend together. But here, since both alleles are dominant, we see both traits. So that's why the dominant red and the dominant white come together to make a flower that shows both red and white. It's the same thing with this particular type of cow. I don't remember, I'm not an agriculturalist or a farmer or anything like that, a bovine expert. I think it's called being a red roan, but don't quote me on that. But the fur color, the patchiness of the white and the brown, the reddish brown, is actually a result of codominance. It's showing the reddish brown allele, which is dominant, and it's showing the white, which is also dominant. So both of those alleles together, you get a cow that shows both of those different fur colors independently. They're not mixed. You end up more with this splotchy, kind of spotted pattern, as you can see in both the flower and the cute little cow right there. Humans also show this. Now, the one example that is the most well-known is blood types. That's actually an example of human codominance, because, and the reason why we say is most people have heard of blood types and know a little bit about them. You watch shows on TV, they talk about, like the crime shows, they sometimes bring that up. Also, since we're in a high school, we have the blood drive twice a year. But your blood type, now we're just talking about the type. We're not talking about the Rh factor, which is the positive or negative. But there are 4 blood types that can be found in humans. You can have blood type A, blood type B, blood type O, or blood type AB. So AB is actually codominant. If you are, or if you possess the A allele and the B allele, then you will show both expressions. You will be blood type A and B. We do have a video, one of my Science in Pajamas videos that deals with blood type, so I definitely recommend checking that out for a little more clarification as how that works, although we will be coming back to it in a little bit because it also has another case or another mode of inheritance. But again, the difference between incomplete and codominance, these are the ones that kids generally confuse the most because they sound very similar. Incomplete, neither allele is dominant, so they blend. Codominance, both are dominant, so both are expressed independently, meaning we see both of the expressions, such as the flower that's red and white, versus incomplete dominance, where we have the flower that ends up pink. All right. So now let's go to multiple alleles. Up to this point, we've only been talking about 2 possible alleles for any given trait. So when we were talking about eye color before, we mentioned the brown and the blue allele. When we were talking about the flower color, we were talking about red versus white allele. But it's not always that easy. It's not always going to be just 2 types of alleles. And we actually see this very commonly in humans, that there are more than 2 types. And we call this multiple alleles. So what we mean by that is that there are more than 2 possible alleles in a population. Now, each individual in the population is only going to possess 2 of the possible alleles, because remember, we have 2 alleles, because we have 2 copies of every gene, because we have 2 copies of every chromosome. So we will only have 2 alleles, but that doesn't mean that there are only allele this and that. What if we have possible alleles this, that, and that? So this way you can have this combination of these 2, or these 2, or these 2, or these 2. What I mean by that is, let's look at blood type. I told you we were coming back to it. So there's actually 3 possible alleles for blood type. And that's why we're able to have 4 types of blood. Remember, we said that for blood type, there's blood type A, B, AB, and O. So how that works is A and B alleles are codominant. And the O allele is actually recessive to both A and B. So if you have 2 A alleles, or you have A and the recessive O allele, you will be blood type A. If you have 2 B alleles, or B and the recessive O, you will be blood type B. If you have an A and a B allele, you will be blood type AB. And if you have 2 of the recessive O alleles, you will be blood type O. Again, we're just talking about blood type. We're not getting into the Rh factor, the positive or negative, because that's controlled by a completely separate gene. So as a result, you can have 1, 2, 3, 4, 5, 6 different allele combinations from these 3 alleles. Homozygous A, heterozygous A. Homozygous B, heterozygous B. Heterozygous AB, or homozygous O. Now, again, everyone only has 2. They either have AA, AO, BB, BO, AB, or OO. But because that there are more than 2 alleles, that can amount to larger possible combinations. We do have a video that talks about multiple alleles and blood type. We kind of go into what's different about them. And that would probably be a good one if you're still curious, still not quite sure what we mean by multiple alleles. I'll definitely go out and check that video out. It's a Science in Pajamas. A link to it will also be at the end of this presentation. All right. Polygenic traits. Now, a lot of what we saw with Mendel, he was just looking at, again, single gene inheritance. So one gene affected the expression of one trait. But what if multiple genes affected the expression of one trait? And we actually see this very, very commonly in humans. So what this means is that there are multiple genes that are affecting how we express a single trait. They can be located on different chromosomes, which is why we see such wide expressions for them. Now, the kind of cool thing about this is because of the wide range of expression, if you were to, let's say, human height, that is a polygenic trait. There are multiple genes that control how tall you are. And if you were to take everyone in a classroom and line them up by height, you would actually see this nice bell-shaped curve, more or less. And that's just because of the sheer extreme. You don't have as many people that are super duper short and a whole lot of people that are like super duper tall, but you have a lot of people in between. So what happens is each of the genes add a small but additive effect. So for instance, with height, the more dominant alleles you have in these genes, the taller you're likely to be. Another example of this would be human skin color. So human skin color is actually controlled by 4 different genes. Now we see more than just 2 skin colors. It's not just dark and light, but then what kind of variation? You have all the way from albino all the way to the other end, which is so dark that the skin appears to be like legit black. Like if you look at Shamar Moore's shirt, kind of like that right there. We see a wide range of skin tones. We see olives, we see pale brown, we see dark brown, we see medium brown, we see tan, we see peach, we see all these different shades. And the reason for this massive amount of variation is simply because of we're looking at how 4 genes are affecting the expression of this one trait. And not necessarily human skin color, but a lot of these polygenic traits, again, it's not just going to be dominant and recessive. You can have codominance mixed in there. You can have some of these polygenic traits, some of the genes in them might be codominant or incomplete dominant. There could be multiple alleles. There could be any number of differences. So then looking at how these incomplete or codominant plus we have something called epistasis, which we're not going to get into in biology, but it's there. Sex-linked versus multiple alleles. And we see that there's a huge variety of different allele combinations between these multiple genes. And that's why we see the huge variation within us for skin color or height or things like that. And here we have just 4 examples of different shades of skin color. All right, the next thing we are going to talk about is called sex-linked traits. Now, sex-linked traits mean that they are located, these are genes that are located on the sex chromosomes. It does not necessarily mean that it has anything to do with the sex of an individual. It may have 0 to do with that. But what it means is simply that the gene itself is located on the sex chromosome. So it actually can be located on either the the X chromosome or the Y chromosome. However, with sex-linked traits, or specifically Y-linked traits, they are super duper rare. As you can see in this picture here, we have an X and Y chromosome. So the pinkish reddish one, that's the X, and the smallish blue one is the Y. There is a clear size difference between the two, and there are far less genes on the Y chromosome. Now, I'm going to say something. I know I say it in one of my other videos, but I always have to say it. Hear me out before you guys get mad at me. There are no life essential genes located on the Y chromosome. How do I know this? Because half the population does not possess a Y chromosome, and yet they still live. Now, that doesn't mean that the Y chromosome isn't important. It is, especially since we are a sexually reproductive species. But what I mean about that is not everyone has a Y chromosome, so therefore there can't be any genes on it that are essential for the survival of that individual organism. Survival of the species, yes. Survival of that organism, no. And since there are far fewer genes located on the Y chromosome, then the chance of having a Y-linked disorder is pretty rare. And again, it'll only affect half the population because females generally will not show it unless they already have some chromosomal abnormalities, like if they are XXY. So we have that going. Typically, when we talk about sex-linked traits, we do talk about what we also term X-linked traits. So ones that are located on the X chromosome, because everyone has an X chromosome, males and females. And so we can see this a little bit more commonly. I'm not saying that they're super common, but they're definitely not as rare as Y-linked. So they code for what we call sex-linked traits, or these are sex-linked genes. We call them sex-linked traits. We simply call them that because they're located on the sex chromosomes. They might have nothing to do with the sex of the individual. There are genes on the X chromosome. They have nothing to do with the sex of the individual, nothing to do with sexual maturity, reproduction, organs, etc. that actually do have life functions on them, which we'll get to a couple examples in a little bit. But we also do notice a unique pattern of inheritance with sex-linked traits. And the reason for that being how our different sexes in humans, how we are chromosomally. Remember, in humans, females have 2 X chromosomes, but in males, you have 1 X and 1 Y. So because of that, we tend to see very unique patterns of inheritance occurring. All right, so what we mean by that is sex-linked traits are more often expressed in males. As you can see here, girls have 2 X chromosomes. So that means that, now typically these sex-linked traits, especially the ones that are disorders, are, what do we call them? Recessive, sorry. So that means that for a girl to show a sex-linked trait, she has to have 2 recessive alleles, 1 on each X. So that's not as common because with females, you can have 2 dominant, 1 dominant, 1 recessive, which we call being a carrier because the girl carries that recessive allele, but she does not show it. She does not express it. Or she can have 2 recessive and be whatever expression that particular sex-linked trait is. However, if you look here, males, or the boy, 1 X and 1 Y. Since the boy only has 1 X chromosome, he will have whatever is located on that chromosome. He will express whichever version is located on that sex chromosome or on that X chromosome. For instance, we mentioned that females can be, let's say, 2 dominant or normal alleles, or 1 dominant and 1 recessive. So a carrier. Either way, she's not going to show the trait. If a guy has 1 dominant allele, the one on his X, he will not show the trait. But if the guy has the recessive allele on his X, he will show it. As opposed to females, which you need 2 recessive alleles to show it. And the reason for this being is that the boy, he is XY. And the X and Y chromosomes are not homologous. I mean, we pair them together because they're both sex chromosomes, but they're actually not homologous. They do not have the same genes located on them. Therefore, whatever is on the X of the male will be what's shown, whether it is 1 dominant allele or 1 recessive allele. He is going to show it. Now, as we said, females, they have two X's, so they have a 2/3 chance of not showing it. They can be homozygous dominant or they can be heterozygous as a carrier. The only way for females to show it is if they have two recessive alleles, whereas males is kind of 50-50. Either they have it or they don't. There we go. All right, let's look at some examples here. These are two of the most common examples that we like to discuss involving sex-linked traits. The first is hemophilia. It is a blood disorder where your blood lacks a certain coagulation factor, which is a protein that helps your blood to clot. As a result, it takes much longer than normal to clot, and this can be very, very dangerous. Most of us, we fall, we scrape our knees, it bleeds for a while, hurts like heck, and then, we'll bandage it up, but it'll eventually scab over. Think about it. What if it doesn't scab over? The point of the scab is twofold. One, it keeps bacteria and other infectious material out, but it also keeps all the rest of the blood in. If you don't have that scab, then the blood will just keep flowing out and out and out, and it can become fatal. So that's why hemophilia is quite dangerous. Now, thankfully today, with lots of medical research, there are treatments where people can get transfusions or supplements that will put the coagulation factor into them. However, their body still doesn't make it naturally on its own, so they always have to go back and get more of these treatments or infusions. The picture on the bottom left, right there, is showing a child with hemophilia who got a bruise. Now, a bruise is simply you fall, you hit your arm or some other part of your body, and it doesn't break the skin, but it still breaks the blood vessel. So what you have is blood pooling underneath the skin. That's what a bruise is. Now imagine, for most of us, again, it'll bruise up, but then eventually it'll kind of start getting pale and it'll disappear. That's because your body fixed the break in the blood cell or the blood vessels, and then this blood that was leaked out just kind of gets broken down. But what if you can't fix those broken vessels? You'll just essentially bleed to death without having a cut in your skin. You just bleed out internally. So again, this can be very, very, very dangerous. And this blood clotting allele is located on the X chromosome. Another one is color blindness, specifically red-green color blindness. Now, what that means is that if you have the red-green colorblind allele, it's recessive. Both of these are recessive. And what happens, is that someone with this disorder cannot distinguish between the colors red and green. And that includes different shades of them as well. So what happens is, it just kind of shows up as shades of gray for both. Now, the bottom right picture is actually a type of colorblind test. Many of us, if we do not have colorblindness, we can see that most of the circles are shades of green, but then inside there are shades of red that make up the number 8. So if you can distinguish between that, if you can see that 8 in the middle, then you are not colorblind. But there are people who just look at that and they just see different shades of gray circles. And that's someone who would be red-green colorblind. Again, it's located on the X chromosome. And, sorry. It's a little early, so I apologize. And with both of these traits, we do see that more often it is men or males that are being affected than females, simply because guys Males only have one X chromosome. So whatever is on that single X chromosome, they're going to show. If they have a normal allele, great. If they have the recessive allele, that means they can end up being colorblind. If a female has, let's say, color blindness, that means she had to get two recessive alleles because she has two X's. Females are less likely to show these traits, not impossible, just less likely, simply because she can either be homozygous for two normal alleles, or she can be heterozygous as a carrier and not show that trait at all. All right, so that's that. We got some additional resources. I would definitely recommend you checking them out, and definitely check some of them out today. Let's see. With the Science in Pajamas, I have a video that compares and shows the differences between incomplete dominance and codominance. It includes Punnett squares of both of those. So I definitely recommend checking those out, seeing the difference, helping you to understand how to do those Punnett squares. Yeah. How do I go back? Oh. No. There we go. I also have a video on multiple alleles, specifically using the example of human blood type. So we talk about what the alleles are in blood type, how codominance comes into play, and the difference between the codominant A and B alleles versus the O allele, as well as what that means for your blood. So definitely maybe check that one out as well. We also have a discussion on polygenic traits and how those have an additive effect to create a spectrum of different expressions for that trait. So that'd probably be a good one to check out as well. I also have an example of sex-linked traits, how we write them out, how we do Punnett squares for them. The difference between the Punnett squares is how we write the alleles. As you can see, I have X and Y's up there. How we typically do the alleles for Punnett squares regarding sex-linked traits is we have the sex chromosome. So we'll actually write out as X and Y, and then we'll put a superscript on the X to show the allele that is located on the X. So I definitely recommend checking those out. They will help you in terms of knowing how to do these in Punnett squares, how to analyze the Punnett squares, and give further descriptions on what exactly is going on. We have some other resources as well. We have Amoeba Sisters, where it looks at codominance, incomplete dominance, polygenic traits, and epistasis, epistasis. And then we have another Amoeba Sisters video looking at multiple alleles. It looks like they focus on blood type as well, including a discussion on how to do the Punnett squares. And we also have an Amoeba Sisters video on Punnett squares and sex-linked traits. Again, these are all great resources. You can check mine out. You can check out the Amoeba Sisters. Theirs will definitely be shorter than my videos because I go into like a lot of detail and explanation. They do a great job of just breaking it down to what you need. So to finish up this lesson, I would recommend strongly that you go check out at least 2, if not more, of these videos today to finish up your lesson. Pick some of the topics that you might not be exactly sure about, check them out. In all the videos I do, I give Punnett square examples, so definitely please check those out. In the meantime, if you have questions, hit me up in our Google Classroom or through email. I hope this helps. Hopefully you guys are doing well, and if you have further questions regarding any of this or regarding, just class in general, please let me know. I can't help you with your questions if you don't tell me you have questions. But in the meantime, Take care, stay healthy, and stay safe. All right, you guys. Adios.